is 4.1.3.3. I really should write them up here. 4.1.3.3, which is transcription in a nutshell. Okay, so transcription is basically where we're making a photocopy of the DNA gene, we're making a photocopy, but that photocopy is not using DNA nucleotides. Remember, DNA has deoxyribose in its structure. We're using RNA nucleotides instead, which are ribose sugar instead of deoxyribose sugar. That's the only difference in their structure. They look exactly the same except for that one little oxygen missing. So transcription is making the photocopy. Now that photocopy can either then turn into functional RNA, or it can turn into, or it can be mRNA, which is messenger RNA, which then goes on to be translated into a protein. Okay, we have a whole heap of different little steps here. You don't need to know much of these steps, so I'm just going to cut them down and give you the very bare basics of them, because you don't need to know them all. But essentially the first thing, and you might have noticed this in your Quizlet, was that you need to understand about RNA polymerase. There is an awful lot of polymerases around. Polymerase means poly, which means you put together pieces or little monomer sections and make a polymer, which is lots of little subunits joined together. And the A's there tells us that it's an enzyme, okay, that's doing the job. So it's an enzyme that's putting together lots of little pieces of something. What little pieces is it putting together? Well, the first part of the name tells you there. It's putting together pieces of RNA, okay? So there are several steps that we need to know when we photocopy or a gene from the DNA. First of all, you have to have that little gene exposed and ready to be copied. Now, there's a whole, I could go into this for hours if I really wanted to, and I don't want to. It'd be way too hard for you guys, but basically just because the gene is there doesn't mean it's going to get copied. The body actually has to turn it on and say, here, I need this gene right now. I have a lack of it out here. You need to actively get in there and make a copy of it. And if you're really low, they'll make multiple copies of it so that you get multiple copies of whatever functional molecule you need. Now, those little signals to turn that gene on, to make it activate and be ready to be photocopied, are called transcription factors. Okay, now there are transcription factors that will join onto the DNA. There are transcription factors that will join onto the histones. There are transcription factors that will join way up here on the DNA and still affect this gene down here. And there are transcription factors that will actually just bind right in front of the gene here and either turn it on or off. There are lots of transcription factors. You don't need to know them all. Be very grateful you don't need to know them all, because that's something that you'll actually do in first and second year uni. And it's changing all the time. I've literally had to throw out my textbooks on this from uni because they're outdated. Okay, but you just need to know that there are transcription factors that either bind to the DNA or the histone factors to either activate them or suppress them from photocopying that gene. Okay, so we've got on the actual DNA here, we've got these little sections here which are promoter and enhancer regions. That's one of the places those transcription factors bind, and they'll either allow this RNA polymerase to join and photocopy, or they'll block it from joining and photocopying. Also down here on the other end, you can even have silencer factors here which will stop the RNA polymerase essentially from joining. That's what they do. If the RNA polymerase can't join onto this little bit here right in front of the gene, so here's the start of your gene, the RNA polymerase has to actually join on right here on what we call a starting section here. I'm not going to go into it in detail, but there's a start code there. The RNA polymerase needs to join there before it can unzip the DNA and photocopy it. If it can't join, nothing happens. So all of these guys basically affect whether or not RNA polymerase can get going. Once he does join on, if he actually does join on here and get ready to go? Well, he actually has to do a few things. The very first thing he needs to do is unzip the DNA so that he can access one of the strands. Okay, because remember there's a double strand here, and only one of them actually has the instructions on it. The other one is just a mirror image and pretty much useless, other than being a mirror image of the gene. Okay, so it's really important that you understand that. I'm going to show you what I mean in just a minute, but first we'll just go through the bare bones here. So he unzips the DNA, then he actually joins on to the nonsense strand, the complementary strand, the one that doesn't have the gene on it. We'll find out why in a minute. He joins on to the opposite side of the gene. So if the gene is here that we need to copy, that's a really bad G, RNA polymerase joins on here, okay? He then goes along and photocopies the gene, okay, and makes a copy using RNA bases, and it could be mRNA or it could just be a function on RNA. So I'm just going to put here RNA copy. So he makes an RNA copy, and then behind him, once he's copied it, he zips up the DNA again. Now that's really important because it's that double helix structure where they're joined like this that protects the DNA from being degraded. When you unzip it, you essentially make it a little bit open to attack. So it has to be zipped up again as soon as possible. Now, once he gets to the end, there's actually a little stop codon that says, "Hui, stop, release," and he does. The RNA then goes off, releases, and lets the little section of RNA that he's photocopied float away. And that little section of RNA that he's photocopied can either fold up into a functional RNA molecule, or it will then be spliced and diced. We'll find that out in a minute. And then to form what we call mRNA, messenger RNA. And then that will be sent outside the nucleus to be translated into a protein. Now, a few things here. Remember how I said that RNA polymerase actually joins on the copy of the DNA that's the gene isn't on. And you might think, what the heck is it thinking? Well, it actually makes an awful lot of sense. Here I've got two pieces of DNA, okay? We have this section here that we want to make a copy of, okay? I need that actual sequence, okay? So I need GACT, GCCT, that sequence. What the RNA polymerase does is that it unzips the DNA like this. And on this side here, this antisense strand, it's a mirror image of the sense strand. Now, if it's a mirror image, remember when we join these little bases up, they're only going to, these floating around in the nucleus here, we have little RNAs just waiting to find a partner. We have G's, A's, C's, U's, all floating around waiting to find a partner. The only thing they're going to partner up with is their complementary base pair. C's will only join up with G's, and A's, because it's RNA, will only join up with U's. So if we want to get a strand that says GACT, GCCT, we actually have to use the mirror image of that strand to get a photocopy. So when they unzip, so there's it's unzipped, and the RNA polymerase joins up here and moves along, he's exposed this C and T and G and A on their own here. Now I'm just going to rub out some of my scribble so that you can have a look at it a bit better. So he's exposed these guys. Well, this G here will come along and he's not going to join up with another G down there. He hates himself. He will join up with a C. So he'll go, ho, okay, there's a C there just waiting for a partner. So he'll actually link up with that C. The A will link up with that T. C will then join up with that G because it's unzipping as it goes. A uracil will join up with the A. I should put 3 little hydrogen bonds there, 2 there, 3 there, 2 there. A G will join up with the C. A C will join up with the G, and so on and so forth. Now look at the order that we've got now. All of a sudden, we have RNA in the same order as the gene we're trying to copy. G, A, C. Instead of a T, I've got a U. That's okay. GCC, GCC, and it works on that way. So by using the mirror image on the antisense strand, we actually end up with a perfect copy of the sense strand. That's why it works. So the promoter region that the RNA joins up to, it's actually here on the antisense strand, not on the sense strand. Okay, because we're actually going to use the antisense strand to make a copy of the gene on the sense strand. So that, I hope, makes that a little clearer. You might actually have to watch that several times to get your head around it. Honestly, that is something that a lot of people struggle with until they really think about it. Okay, in order to make a copy of this sense strand, you have to use its mirror image. Okay, it's sort of like, in order to make a copy, if you're someone who's making like a bowl or something, by taking like a plastic impression of it, like a hand or something. If you're making an impression of someone's hand, the first thing you'll do is make a mirror image of that hand by putting their hand in like plaster or foam or something and having it solidify. But then you have to then use that mirror image of that hand to then fill it up with plaster to make then a copy of the hand shape itself. So when we do impressions in plaster and stuff for art, we are literally making a mirror image of what we need to make a copy of. And DNA, RNA works exactly the same way.